Okay, I want to start today with a little mental exercise. I want you to close your eyes for a second, unless you're driving. Please don't close your eyes if you're driving. But picture a mushroom,
Mhm.
specifically a poisonous mushroom, just the first image that pops into your head.
Oh, yeah. I think I know exactly what everyone is picturing. It's got that red cap, right?
It is always the red cap.
Bright red, white dots, a little white stem,
Yeah,
the classic Amanita muscaria, the fly agar. Yeah.
exactly. It's the emoji mushroom. It's the little house in the Smurfs. It's the power-up in Mario.
Hmm.
It is uh, the absolute postcard child for the entire fungal kingdom.
It really is.
If you ask a kindergartner to draw a toadstool, that is what they draw.
It's iconic. It's the celebrity of the fungal world, no question.
But here's the thing, and this is really the hook for what we are diving into today. That image is kind of a lie. Well, maybe not a lie, but it is a massive distraction.
Hmm.
It represents the flashy, the photogenic minority of the whole genus.
It really does. It sucks all the oxygen out of the room
Yeah.
because the reality of the forest floor, especially if you are walking through woods in, say, Europe or really anywhere in the Northern Hemisphere, is it's very different.
Right. You aren't tripping over red and white toadstools every 5 feet. Not at all. You are seeing something else entirely. You are seeing the hidden majority.
You're seeing hundreds, maybe thousands of mushrooms that are, well, they're gray or
Yeah.
brown or beige or a sort of nondescript grise.
Refuge. I love that. The interior design color of the forest.
Exactly. They are tall, they are graceful, they are absolutely everywhere, but they are frustratingly hard to tell apart. For a long, long time, we just called these the Ringless Amanitas.
The Ringless Amanitas.
Mhm.
Or, to be a bit more scientific, Amanita section vaginate. And today, we're going to talk about why this specific group of boring-looking gray mushrooms is basically the final frontier of mushroom classification.
It's a detective story, really, a molecular one.
It really is.
Yeah.
We have a massive study from 2024, so fresh off the press, published in the journal Mycological Progress. It's by Dora Varga, Jean-Michel Hunt, and a whole team of colleagues. And what they did was, well, they basically took the trash can of gray mushrooms and dumped it out on the table to see what was actually inside.
Yeah, and they didn't just look at them, they looked at their DNA. And not just one gene, but 4 different genes. And they looked at the microscopic architecture of their tissues in a way that really hadn't been done with this kind of rigor before.
And the result?
The result is that we have been walking past species for decades, centuries, even though we thought we knew, but
Right.
he didn't have a clue. They discovered 3 brand new species hiding in plain sight in European forests.
Hiding in plain sight. I just love that concept. We're going to meet Amanita deflexa, Amanita grisufulva, and Amanita opaca. 3 new characters that have just entered the chat.
And we're also going to look at how this changes the game for identification. I mean, we're moving from it looks gray, so it must be this, to let's sequence the genes and measure the geometry of the spores. It's a whole new level.
We're also going to bring in a field guide entry from First Nature on a species called Amanita submembranatia, just to contrast the sort of old way of identifying things with this new high-tech approach.
It's a perfect example of how the field is evolving. It really is.
So our mission today is to explore this hidden majority, to understand how modern science is splitting the atom, or splitting the mushroom in this case, to reveal a level of biodiversity we never knew existed.
It's going to be a deep dive into the details, but I promise the details are where all the magic is.
Absolutely.
Yeah.
So let's set the stage. We keep saying section vaginate, keep saying ringless amanita scos, but what does that actually mean? If I'm a listener walking in a forest in France or Germany or even here in the US, how do I know I'm looking at one of these?
It's a great question because the genus Amanita is huge. I mean, there are about 1,000 estimated species worldwide, but section vaginate, the ringless ones, they make up a huge chunk of that. Maybe 400 species?
400. That's nearly half the genus. That's wild.
It is. They are incredibly successful, evolutionarily speaking. To spot one, you really just need a checklist of 3 things. It's like a diagnostic triad.
Okay, hit me with the triad. What's number 1?
Number 1 is the cap margin, the very edge of the mushroom cap. In this group, it is almost always what we call striate.
Striate. That means it has grooves, right? Like little lines.
Yes, exactly. Think of radial lines extending from the edge of the cap towards the center. It's almost like the gills underneath are imprinting on the surface of the cap.
Oh, that's a good way to put it. Like you can see the skeleton through the skin.
A little bit. It's like the ridges on the side of a quarter or like the pages of a book fanned out. If you run your finger along the edge, you can feel those tiny ridges. That's a huge clue.
Okay, so I've got a mushroom with a grooved edge. What's number 2?
Number 2 is right there in the name. They are ringless.
Which is what distinguishes them from the famous ones, like the fly agaric or the death cap.
Right. If you picture a death cap, they usually have that skirt or a ring on the upper part of the stem. We call that the annulus.
That's the leftover bit of veil, isn't it?
It's where the partial veil broke as the mushroom opened. Section vaginata doesn't have that. The stem is usually long, slender, and clean. No skirt.
So it's naked.
Basically, yes. A naked stem, no annulus.
And the third thing, you said it was a triad.
The third thing is the vulva. And this is crucial. This is the feature we are going to come back to again and again and again today.
The vulva is that sac at the base, right? The thing it hatches out of.
Exactly. At the very base of the stem, and you often have to dig a little, it's usually buried in the dirt or the leaf litter, there is a sac-like structure. It looks like a broken eggshell that the mushroom just burst out of.
Because that's literally what happened.
It is. It's the remains of the universal veil, the tissue that covered the entire mushroom when it was a baby egg. As it grew, it broke through the top, leaving the sac at the bottom. In section vaginatae, that sac is usually very, very prominent.
Okay, so let's recap the trinity. We've got a striate cap, meaning a grooved edge.
Yep.
We have no ring on the stem.
You're right.
And a big sac-like vulva at the bottom.
That's your trinity. If you see that combination, you are almost certainly looking at a ringless amanita, or what foragers often call a grisette.
Sounds easy enough to spot the group then. But here is the tension, and this is the problem that led to this whole study. If they're so distinct as a group, why has identifying the specific species inside that group been such a complete nightmare?
Because of a concept we call cryptic species.
Cryptic species.
It's the idea that different species can evolve to look nearly identical. Nature finds a body plan that works. This tall, slender, grayish form is apparently very efficient for spore dispersal in these forests. So multiple, completely separate genetic lineages settle on that same look.
It's convergent evolution. Like how sharks and dolphins both ended up with fins and a streamlined shape.
Exactly. They're wearing the same uniform. So for the last 100 years, if a mycologist in Europe found a gray, ringless Amanita, they would just shrug and say, it's Amanita vaginata.
It became a catch-all term, a default.
Total wastebasket taxon.
Yeah.
If it didn't fit anywhere else, you just threw it in the vaginal bucket. If it was a bit reddish-brown, maybe you'd get fancy and call it Amanita fulva, the tawny grisette. But that was about as granular as it got.
So the just look at it method completely failed.
It failed because our eyes literally cannot see the difference. They are too subtle.
Yeah, that brings us to the 2024 study by Varga and her team. They decided that good enough wasn't good enough anymore. They wanted to really know.
No, they wanted to know the truth. And to get the truth, you have to go deeper than the skin. You have to go to the DNA.
And they didn't just do a quick swab. This was a phylogenetic study. A serious one.
Yeah, a really robust one. They analyzed 151 different specimens from all over Europe, France, Germany, Hungary, Corsica, you name it. And they used what they call a 4-locus approach.
Okay, let's unpack that. 4 locus. That means they looked at 4 specific spots on the DNA map.
Correct. Usually in fungi, researchers look at the ITS region. That's the internal transcribe spacer. It's like the standard barcode for life in the fungal kingdom.
It's like checking the VIN number on a car to see what model it is.
Exactly. But the problem is, in this group of mushrooms, the ITS can be a little blurry. It doesn't always have enough variation, enough little changes to tell very close cousins apart. So Varga's team added 3 more regions.
Okay.
The LSU, which is the large subunit, and 2 protein coding genes called RPB1 and TEF1 alpha.
That sounds like alphabet soup.
It does, but think of it like GPS triangulation. If you just have one satellite, that's your ITS, you know you're somewhere in Europe. If you add the LSU, you know you're in France. If you add RPB1 and TF1 alpha, suddenly you're standing in a specific driveway looking at a specific house number.
It gives you the resolution to see the separate lineages. It sharpens the image.
It gives you incredible resolution. It clears up all the noise. And when they ran this analysis on those 151 common gray mushrooms, the family tree just exploded.
Exploded how?
They found 36 distinct species lineages just in that single sample set.
36. From 151 mushrooms, and we thought there were just, a handful of species.
Right. And amongst those 36, there were 3 clusters that didn't match any known name in the databases. They were genetic orphans. And that is the smoking gun for a new species.
So discovery in 2024 isn't about hacking through the jungle with a machete anymore. It's about running an algorithm on a sequence alignment in a lab.
Well, ideally, it's both. Because, and this is what I love about this paper, it's so thorough, they didn't just stop at the DNA. They didn't just publish a spreadsheet of gene sequences. They went back to the physical mushrooms.
Right.
They said, okay, the DNA says these three groups are different. Now let's go back and look at them. Can we find a physical feature, something we can see that matches the DNA?
They went old school.
They went back to the microscope.
Which brings us to section 2 of our discussion, and this is my favorite underdog story of the entire episode, the spore.
The humble, overlooked spore.
Yes. I was reading the background for this, and apparently, for a long time, mycologists thought that measuring spores in this particular group of mushrooms was a complete waste of time.
It was dogma. The prevailing wisdom, the thing you'd read in old books, was don't bother measuring spores in Amanita section vaginate. They are all globose.
Globose meaning round.
Well,
spherical?
Just a ball, and a ball's a ball. If they're all perfectly round, what's the point of measuring them? It was considered a useless metric. People just didn't do it.
I'm guessing that was wrong.
Completely wrong. Varga and her team, they applied rigorous statistical analysis. They used ANOVA tests, which compared the means of different groups, to the spore measurements. And what they found is that while they all look round to the naked eye under a microscope, there are statistically significant differences.
So one species might be, say, 8 microns on average, and another might be 8.5 microns.
Exactly. Or, even more importantly, one might be slightly more oval. They looked at something called the Q value, which is the ratio of the spore's length to its width. A perfect circle has a Q value of 1.
I see.
They found that one species might have a Q value of 1.05, and another has a Q value of 1.1. And even if that difference is tiny, if it is consistent across dozens of specimens, it's a valid identification feature.
So the spores do tell a story. We just weren't listening closely enough. We weren't using the right tools to listen.
We were too lazy, or maybe just too confident in our old assumptions. But this really matters because it empowers the field mycologist. It means you don't always need $1 million DNA sequencer in your backpack.
Right.
Sometimes, if you are very careful and you have a good microscope and you know how to do the math, you can identify these species based on the spores. It brings the science back to the bench.
I love that. It's a victory for old-fashioned, careful observation, but backed up by modern stats. Okay, so we have the method. High-tech DNA plus high-stats microscopy. Now let's meet the stars of the show. The 3 new species that this study officially named and described.
Let's do it. They're fascinating.
First up, we have Amanita deflexa.
Amanita deflexa.
I have to say, deflexa sounds like a spell from Harry Potter. Deflexa.
It does. It's actually Latin for to turn away or to move away.
Why that name? Is it antisocial? Does it grow away from the other mushrooms?
In an evolutionary sense, yes. It was named that because historically, this mushroom was constantly confused with the tawny grisette Amanita fulva. They can look very, very similar. But genetically, Amanita deflexa has turned away from Amanita fulva. It has diverged onto its own path.
So it's the rebellious cousin. I like it. Let's paint a picture. If I'm in the woods, what am I seeing?
You're seeing a mushroom that starts out quite dark. The cap is bell-shaped at first. We call that campanulate. And then it flattens as it gets older. The color is... well, it's tricky. That's the problem. It starts dark, then turns a sort of yellow to orange, but then it gets dull and opaque as it ages.
That sounds incredibly vague. Yellow to orange, but sort of dull. I feel like I've seen 1,000 mushrooms that fit that description.
You have, and that's precisely why it was missed for so long. If you just look at the cap color, you'd walk right past it and say, oh, that's a tawny grisette, and move on. But there is a huge clue, and it's ecological.
It's the trees. It's always the trees.
The trees are everything here. Amanita fulva, the classic look-alike, usually loves deciduous trees, oaks, birches, beeches, that sort of thing. But Amanita deflexa, it is strictly associated with pines.
The tree, as in 100% of the time.
In the study, every single specimen of deflexa that they sequenced and confirmed was found with Pinus sylvestris, which is Scots pine, or Pinus nigra, which is black pine.
Okay, that is a massive field marker. So if I'm an amateur mycologist and I'm standing in a pure pine plantation and I see something that looks like a tawny grisette, I should pause.
You should definitely pause. Your alarm bells should go off. You should think, wait, this is the wrong tree for a tawny grisette. What else could this be?
But the real smoking gun, the thing that makes this a slam dunk new species scientifically, is not something you can see from a distance. It's under the microscope. It's in that sac at the base, the vulva.
This is the part of the study that really impressed me, the level of detail. They dissected the vulva tissues. And for Amanita deflexa, they found a distinct two-layered structure.
2 layers.
Yeah.
Like a jacket with a fancy lining.
Exactly. That is a perfect analogy. The outer layer is what we call filamentous. It's made of long, thread-like hyphae. But the main inner layer is made of these physiloid cells.
Physiloid. That's a great word. What does it mean?
It means inflated and bubble-like. So imagine big, round balloon cells making up the bulk of the tissue.
And the color. The color was the giveaway, wasn't it?
The color is the kicker. So the vulva is white on the outside, just a plain white sac. But if you look at the inside surface, or the upper rim where it tears, it is strongly colored, a deep red-brown to yellow.
So it's a white bag with a rusty orange lining.
Precisely. And here is the significance. According to this study, Amanita deflexa is the only European taxon in this specific group, they call it the fulvostirps, that has this specific two-layered anatomy with the colored inner surface.
That's definitive. That's not a judgment call. That's a structural fact.
It is. It's a structural blueprint that is unique to this species. So if you have a microscope, you can see this. But even if you are just in the field, you can look for the pine trees and then carefully check for that colored lining on the vulva. It might be all you need.
It's a perfect example of how specific these organisms are. It's not just growing on the ground. It is partnered with a specific tree, and it builds its protective shell in a completely unique and identifiable way.
It really makes you appreciate the complexity that's been hiding there all along.
All right, that's Amanita deflexa, the pine-loving rebel with the fancy coat lining. Let's move to species number 2, Amanita griseofulva.
Yes, Amanita griseofulva.
Let's break down the Latin again. Griseo means gray. Fulva means tawny.
So gray tawny.
That sounds like a contradiction. Is it gray or is it tawny?
Yes.
Thanks, that's very helpful.
That's the problem. The name actually describes the confusion perfectly. This species looks like Amanita fulva. It has that slender, graceful shape, that tawny color, but it has a distinct grayish cast to it.
The study calls it a chameleon, which suggests the color is all over the place.
It really is. The cap can be gray, gray-beige, yellow-gray. They even found some collections that were dark gray-brown or had a bronze-green tint to them.
Bronze green. That's a lovely image. But again, it sounds like color is really variable. If I can't rely on color, what are the hard details? What should I be looking for?
Okay, there is a very cool feature on the stem. On young, fresh mushrooms of Amanita griseofulva, you can see these tiny white aggregates.
Aggregates? What does that mean? Like little crumbs?
Yes, exactly like breadcrumbs stuck to the stem. Or floccules, little tufts of white fluff.
Floccules, another great word.
These are remnants of the partial veil. Now remember, we said this whole section is ringless. They don't have a partial veil that forms a sturdy ring like a death cap, but they do have traces of it. And in this species, those traces appear as these little white clusters that eventually wash away with rain.
So that's a detail you have to look for on a fresh specimen. Little ghost crumbs on the stem.
Exactly. And then again, you look at the vulva. Unlike the sturdy double-layered sac of deflexa, the vulva of griseofulva is described as vaginate, fragile, and rust-stained.
Rust-stained. That's memorable, like it's been sitting on old metal.
It often has these little reddish-brown spots that develop, especially as it ages. And because it's so fragile, it often breaks into small patches rather than staying as a nice, clean cup at the base.
And there was something about a ghost version of this mushroom in the study, a white one.
Ah, yes. They formally described it, Amanita griseofulva form albida.
Albida meaning white.
They found a version of this species that is genetically identical. I mean, the same DNA barcode. But physically, it is pure white. It's albinotic.
That must be so confusing for a forager. You spend all this time learning to look for a gray, tawny mushroom with rusty spots, and suddenly nature throws a pure white one at you.
It happens more often than you think in fungi. It's just like you have albino animals. But because they checked the DNA, they could confirm with certainty that it wasn't a different species. It was just a color variant of griseofulva.
So fascinating. Now, let's check the habitat. We know deflexa was the pine lover. What does griseofulva like?
It is the absolute opposite. It strictly prefers deciduous forests, oaks, quercus, hornbeam, carpinus, beech, fagus. The study specifically notes that it does not grow with pines.
So once again, the forest type is your first and maybe your best clue. If you're in a pine forest, you're thinking deflexa. If you're in an old oak forest, you should be thinking griseofulva.
Exactly. And genetically, this one is really interesting because it's what we call a basal species.
Basal. That means it's at the bottom of the family tree, an early branch.
It means it branched off very early in the evolution of this group. In the phylogenetic tree of all the tawny Amanitas, Amanita grisofulva is sitting right there at the base. It's separated by a significant genetic distance. The paper says 16 substitutions in the ITS region compared to its closest relatives.
That's a big gap in fungal terms, isn't it?
It is. It's like finding the great-grandfather of the group. It's an ancient lineage of gray tawny mushrooms that's just been quietly hanging out under the oaks for millions of years, and we're just now giving it a name.
All right, 2 down. We have Deflexa and Grisofulva. Let's talk about the third new discovery, Amanita opaca.
Amanita opaca.
The name sort of gives it away. Opaque, not shiny.
Yes, the cap surface isn't shiny or sticky. It has a dull, almost matte appearance.
The description of the color in the paper was interesting. They called it bicolored.
Right. It's usually a mix of gray and another color, like a beige or a brownish tone, with the center being noticeably darker. It creates this gradient effect from a dark center to a lighter striate margin. And the stem is stark white.
Stark white stem. And what about the vulva on this one?
The vulva here is described as having a short collar. It's vaginate, meaning it's sheathed around the base of the stem, but it opens up into a collar shape at the top. It doesn't come up as high on the stem as some of the others might.
Okay, but we have to talk about the microscope part for this one, because this was my absolute favorite detail in the entire paper. The springs.
I knew you'd like this. This is a marvel of microscopic engineering. It's truly beautiful.
Tell the people about the springs.
So when they looked at the tissue of the vulva in Amanita opaca under high magnification, they found the hyphae, the cellular threads, were arranged helically.
Helically, you mean like a DNA strand or a slinky?
A spiral.
Exactly.
Like little tendrils or twists wrapping around those big balloon-shaped physiloid cells we talked about earlier.
That is just wild. Why would a mushroom do that? What's the point?
The extra analysis in the paper suggests this structure acts like a spring to provide mechanical coherence to the tissue.
So the mushroom is building its own internal shock absorbers. Or structural support, like rebar in concrete.
Essentially, yes. Think about what the vulva does. It's the first line of defense. It has to protect the young mushroom as it pushes up through the soil, which can be heavy and compacted and full of sharp petals. By wrapping these helical, spring-like hyphae around the inflated cells, it creates a tissue that is both tough and flexible.
It can stretch and compress without tearing?
Precisely. It's like reinforced concrete, but on a microscopic scale for a mushroom.
That is incredible. You look at a mushroom and think it's just mush. It breaks if you look at it wrong.
Yeah.
But under the lens, it's woven together with these intricate spirals to create strength.
Nature is the best engineer, and this feature is what distinguishes it from its sister species. Amanita opaca is genetically very close to 2 other species, Amanita cystatorum and Amanita corylli. But those species don't have the specific helical structure in the same way.
And again, genetically, it's distinct. The study says it has about 98% similarity to its sisters.
Which sounds incredibly close to a layperson.
Right. Humans and chimps are what, 99% similar in some measures?
Exactly. But in fungi, a 2% difference in these core gene regions is a chasm. It's a smoking gun for a different species. It means they are on distinct evolutionary paths and have been for a long time.
So we have Deflexa, the pine lover with the 2-layered vulva, Grisofulva, the oak lover with the white crumbs and rust stains, and Opaca, the dull-capped one with the helical springs in its vulva. 3 brand new players on the European stage.
It's a whole new cast of characters we didn't even know were in the play.
But, you know, we can't talk about the new players without giving a nod to the old guard. Because listeners might be thinking, are all ringless Amanitas new and confusing? And that's not quite true.
Right. Let's look at a species that was already known, but is rare and fascinating in its own right. Amanita submembranacea.
The olive Amanita.
We pulled a source from the website First Nature on this one to give us some of that classic field guide context.
So how does this one contrast with our new friends? What makes it different?
Well, first, it's an altitude junkie. While our new species are found in various lowland forests, Amanita submembranacea loves the mountains. It grows in high-altitude coniferous forests, spruce, fir, larch.
And the description of the vulva in the First Nature Guide is just. It's poetic. I have to read this quote. They say it looks like flakes of dull gray paint on a canvas background.
Flakes of dull gray paint. That is so evocative, isn't it? It's not a clean white sack like Deflexa. It's gray, it's leathery, and it's often crumbling and patchy.
And the stem isn't clean and white. It has a snakeskin pattern.
They call it zebrina or zonet. It's a much darker, grayer, more rugged-looking mushroom than the delicate opaca or grisofulva. It really looks like it belongs in that harsh mountain environment.
Now, here is where I want to do a quick public service announcement. We are talking about Amanitas.
Yes, the most famous and infamous genus.
Some Amanitas, like the Caesar's mushroom, are choice edibles. Others, like the death cap or the destroying angel, well, the name says it all. Where do these ringless ones fall on that spectrum?
The general rule with ringless amanitas or grisettes is tread very, very carefully. Traditionally, in Europe, some of the common ones, like Amanita fulva or Amanita vaginata, are eaten. People cook them thoroughly and eat them.
WT, I hear a giant butt coming.
It is a huge BUT because, as we have just spent the last 40 minutes proving, identifying them down to the species level is incredibly difficult.
The first nature guide explicitly lists Amanita submembranatia as suspect.
Exactly. Suspect. It means we don't know for sure if it's toxic, and we certainly don't recommend you be the guinea pig. When you have cryptic species that look identical but are genetically different, you cannot assume that because one is safe, the look-alike is also safe.
Toxicology can vary just as much as DNA.
Exactly. A tiny genetic shift that changes a spore shape might also be linked to a gene that produces a novel toxin. We just don't know yet.
So the takeaway is, if you find one of these newly described species like Amanita deflexa, don't eat it.
Please do not eat the scientific discovery. Let it be. Admire it, photograph it, but leave it for science.
So how does this old guard mushroom, submembranatia, fit in with the new guys? The phylogenetic study puts it in its own stoops or group, right? So it's not really related to our new friends.
It's distantly related. They are all in section vaginato, but submembranatia is off on its own branch. It confirms that just because two mushrooms both grow under conifers, like submembranatia in the mountains and deflexa in the lowlands, it doesn't mean they are sisters. They arrived at that habitat independently.
That makes sense. I want to zoom out a bit now. We've geeked out on vulvas and spores and helical hyphae, but why does this matter? What's the so what? Why should the average person care that we just split one gray mushroom into three?
That's the big question. And I think the most important answer comes back to the trees.
The ectomycorrhizal partnership.
Yes. All of these Amanita species are ectomycorrhizal. They are not decomposers.
Can you break that word down for us again? Ectomycorrhizal.
Sure. Ecto means outside, myco means fungus, and rhiza means root. These mushrooms aren't just rotting on the ground eating dead leaves. They are partners. Their mycelium, the underground network, physically wraps itself around the tiniest roots of living trees.
Like a glove on a hand.
A perfect analogy, like a glove. They extend the tree's root system by thousands of times, going out into the soil and gathering water, nitrogen, phosphorus, things the tree struggles to get on its own. And they trade those nutrients to the tree in exchange for sugars that the tree makes from sunlight.
It's the original internet, the wood wide web.
It is the life support system of the forest. So when we say Amanita deflexa needs pines, it's not just a casual preference. It's a biological handshake. It's a marriage that's millions of years old.
And this implies that conserving these fungi requires conserving very specific forest types.
Exactly. You can't just plant trees and expect the ecosystem to be healthy. If Amanita grisifulva relies on ancient oak and beech forests, then losing those specific forests means losing that unique genetic lineage of fungi forever. We are mapping the complexity of the forest floor. By naming these species, we are realizing that the ecosystem is so much more intricate than we ever thought.
This study mentioned a statistic that just made my jaw drop. It said that about 220 new Amanita taxa have been described in just the last 2 decades.
It's staggering, isn't it? It means we are living in a golden age of fungal taxonomy.
A golden age.
Absolutely. For centuries, we were limited by what our eyes could see. We were lumpers. We lumped things together because they look the same. Oh, it's gray. It's a grisette. Now, with DNA sequencing becoming cheaper and more accessible every year, we've become splitters.
We're cracking open the lump.
We're cracking it open, and we are finding that the room is crowded. We are realizing that the biodiversity on the forest floor is orders of magnitude higher than we ever imagined.
It's like we finally turned on the high-resolution camera after looking at a blurry photo for 200 years.
Or just turning on the lights in a dark room, and we're finding that the room is full of strangers we've never met.
I love that image. It also highlights the pace of science. Amanita deflexa was just considered a weird variation of fulva yesterday. Today, it's its own species with its own story. Science isn't a static book of facts. It's a living document that we're constantly revising.
And it requires us to be humble. We have to be willing to admit what we don't know. The more we look, the more we realize how much we have left to learn. It's an amazing time to be studying this stuff.
So let's recap our new strangers for the listener who might be heading out for a hike this weekend. Give us the cheat sheet. the quick and dirty guide.
Okay, here's the summary of our 3 new species.
Number 1, Amanita deflexa.
That's the pine lover. Look for it strictly under Scots or black pine. Carefully check the vulva at the base. It should be a sturdy sac, white on the outside, but with a distinctly colored reddish-brown lining on the inside.
Number 2, Amanita grisafulva.
The gray tawny. You're going to find this one strictly under leafy trees like oak and beech. Look for those little white crumbs on the stem of young ones, and a very fragile vulva that might have rusty spots on it.
And number 3, Amanita opaca.
The dull one. It has a bicolored grayish cap that's darker in the center, a stark white stem, and a short collar-like vulva. And if you had a microscope, you'd be looking for those beautiful spiral springs in the tissue.
And just for contrast, the old guard, Amanita submembranacea.
That's your high mountain specialist, growing with spruce and fir, and it has that unforgettable vulva that looks like peeling gray paint.
You know, this brings me to a final thought. We are talking about Europe here. France, Hungary, Germany. These are some of the most studied ecosystems on Earth. People have been picking and studying mushrooms there for thousands of years.
And yet, we are still finding brand new, widespread species.
Exactly, that's the point. If we were finding 3 new species just by looking closely at common gray mushrooms in Europe, what on earth is hiding in the Amazon? What is hiding in the Congo Basin?
Or heck, what is hiding in a patch of woods in suburban Ohio that no one has ever bothered to sequence yet?
It's mind-boggling. The scale of our ignorance is just vast.
The vast majority of fungi are still unnamed. Some estimates say we have only named maybe 5% to 10% of the world's fungi. The rest are essentially dark matter to us.
Fungal, dark matter. That is both exciting and terrifying.
I think it's an invitation. It means the age of discovery isn't over, it's just beginning. You don't need a spaceship to find new life forms. You just need to look down.
So that is our call to action for you today. The next time you are out walking and you see a grisette or a ringless amanita, don't just walk by. Don't just say, oh, a mushroom. Stop. Look closer. Look at the trees. Look at the vulva. Is it rusty? Is it peeling paint? Is it double layered?
You might be looking at something that has a brand new name, or you might be looking at something science hasn't met yet at all.
You might be looking at science in the making. Thanks for diving deep with us today.
It's always a pleasure.
Keep looking down, everyone. You never know what you'll find.